Good morning, sir. Sir, good morning, sir. Can you hear me, sir?
Yes, Mr. Ram Praveen. Yeah, I
Yes,
can
sir.
hear you. Yeah.
Okay, sir. We'll wait for another 5 minutes, sir, so that they can join. We'll start.
No problem.
Yes, sir. Thank you, sir. Sir, can you able to slide show it,
Anybody
sir?
has to join?
Sorry, sir.
Anybody has to join.
Yes, sir. Actually, we have 60 participants, sir. So
Okay.
right now only 38 join. We'll wait for
Okay.
another 5 minutes so that everybody can join.
Okay, sure.
Sir, is there any difficulty, sir, or can you able to slide show it, sir?
No problem, boss. It's fine.
Yeah, fine, sir. Okay, thank you, sir.
Can you see Can you see
Yes,
this?
sir. We can see your slides, sir, but the slideshow, I think it is not.
No, that I have to make it. Yeah.
Okay, fine.
It is moving, right?
Yeah. Fine, sir. Fine,
Yeah.
fine. Okay, sir. We'll start formally after 10 minutes or 5 minutes.
இது இருக்கு.
Sir, shall we start, sir?
Yeah, you can start, Ma.
Over to EMC, you can start the proceedings.
So can you hear me?
Yes, ma'am, you can start.
Yes, yes.
First of all, I would thank our beloved principal, Dr. M. Ramanathan, to providing me this opportunity and to organize such a fabulous program, scientific program, which will enlighten all participants and delegates. So research is to see what everybody has seen and not to think what nobody else has thought. Good morning all. A very warm welcome to the third day of the refresher course. Today's first session is on the topic hypothesis framing, rationale, aim and objectives. For that, I would like to welcome our respected speaker, Dr. A. Justin. Dr. A. Justin is currently working as an assistant professor in the Department of Pharmacology, JSS College of Pharmacy, OT. He has done his doctoral research in neuropharmacology and had been worked in PhD College of Pharmacy from 2010 to 2015. He had secured gold medal and Excellence in Pharmacology Award from Tamil Nadu Dr. MGR Medical University for scoring first rank in post-graduation in pharmacology. Dr. A. Justin is holding one DST SERB major research of rupees 32 lakhs and 2 minor research grants. He has been guiding 5 PhD scholars and has published 30 international and 10 national publications, along with 2 book chapters. He has filed 2 patents and is a reviewer of various journals in the field of pharmacology, toxicology, and neuroscience. Now let us greet Dr. A. Justin, sir, who will begin today's session with hypothesis framing, rationale, aim, and objectives. Welcome, sir, and over to you, sir.
Yeah, thank you, madam, for your nice introduction. And I profusely thank the organizers, PhD College of Pharmacy, and the respected principal, my mentor, and my ex-colleagues, and and the faculties of Department of Pharmacology, especially, and HODs and research scholars of PhD College of Pharmacy. So thank you for this. Once again, thank you for the good opportunity given. So today I am going to discuss about the writing hypothesis and the objectives of the project. So just an introduction, because nowadays everybody writing the proposals to various funding agencies, and also we are the proposals, not only proposals, we are also designing some of the research protocols for the PhD thesis or MPharm work, dissertation work, and even for the BPharm projects works. So now, what is the basic need while writing the hypothesis or objectives? So let us have a glance. It is an interactive session only. Okay, anywhere you can stop me and we can have a discussion also. So just an introduction before we going for any hypothesis. For example, in the field of with respect to pharmacy, pharmacology or cognizy or pharmacy practice, or other disciplines like biotech, microbiology, and also engineering, whatever maybe. So before hypothesis, we must create our area of research interest. So where we are going to work, so that first we have to decide, and we have to create the science hypothesis. That is very important. Before going for the research hypothesis, we must have the science hypothesis. So there is just a simple difference between the research hypothesis and the science hypothesis. Science hypothesis, or we can see continuous seeing of the monitor and also mobile can reduce the eyesight, vision problem may happen. So that is called the science hypothesis, okay. This is common for everyone. Okay, this is, you can create your science hypothesis anywhere. But a research hypothesis is nothing but because what is the reason the eyesight is losing? So what is the radiation emission from this display where it is, for example, retinal damage or corneal problem or anything else? So what is happening? That is the first general objective of your research hypothesis. Next is, what may be the reason? So what is happening in the cellular level? Any degeneration of the cellular membranes of retinal, and also anything happen to your rod cells or corn cells, whatever maybe. So that is your research hypothesis. So general term is science hypothesis. We must first create, and we have to, from that we have to create the research concept. So what I am going to do. So best example, again I'm saying again I'm quoting this also. What is the mechanism for the vision impairment? That is my research concept. And also research gap. So what is the gap? Why it is happening? So what I can able to fulfill it. Okay, between the previous researcher, what is his concept? So what are my thoughts? What is his gap? What is the justification can be given? And also the applications of the research. So any research, if you are carry out any type of research that may be in any topics, that should be applied in somewhere. Okay, because if the research cannot be applied, for example, application in the form of not only specific to the society, so it may be beneficial to other researchers also, like citations, and also they can follow your methodology, or they can follow your hypothesis, how from that hypothesis, how I can deviate from that, how betterment. Okay, like that. So novel area. So because we know that a lot of work we are doing, okay, most of the works are outdated now. Most of the journals are not accepting. The best example we can say now, the antioxidant assays. So antioxidant assays are outdated now. Okay, and also then those types of the outdated things we can negligent. And the novel area, what is the need now? So for example, I am going to do in the now COVID or any future Omicron or whatever may be. So novel area, I'm just giving the example. It should be latest. It is the need of the hour. Okay, like that we have to take the project and design the before hypothesis, and also feasibility. That is very, very important. See whether I can do this. For example, again COVID. So I'm going to design the drug for the COVID. Okay, you have the computational, you have everything, but where will you check? You have designed the computational method, you have designed the drug, and also you have developed so many molecules also in your synthetic lab. The characterization of using NMR, mass, everything done. But where we are going to evaluate, whether you are having the BSc standard lab, whether we are having the viral culture. So that is very important. So before going for any study, feasibility. So otherwise we have to create the feasibility or arrange the feasibility. So that is very, very important. And research place where we are going to work. For example, if you are designing any protocol, this in vitro I'm going to do here, in vivo I'm going to do here. Before that we have to ensure the workplace also. So these are the points we must understand before going for your hypothesis. Okay, so the area of research interest we should create science hypothesis. From the science hypothesis, we go for the research concept, and the research concept, while developing the research concept, we must know what is the research gap and application of the research to some level, and it should be very novel and it should be feasible to do, and also research place where we are going to the study. So those points we must understand before creating the hypothesis. So next, we are going to see what is meant by hypothesis. Hypothesis is nothing but a hypothesis is a precise testable statement of what the researcher predict will be the outcome of the study. It is stated at the start of the study. So just we are going to create one hypothesis that should be testable. That is very, very, very important. Okay, also you should give the good outcome of the study. Also, after seeing the hypothesis, anybody can come to understand what is your next objective. Okay, what is your outcome? So it will be very, very easy statement. So it should not be very broad, long sentence. So it may not be reach others. So people may avoid the readings of those hypotheses. So it may be failures of that is a major cause of the failures of the research proposal, what we are going to submit in the various funding agencies. A single sentence, a research hypothesis describe the results that the researcher to find. That's all. It is a prediction or an educated guess about the outcome of one expert from the undertaken. So there are two types of hypothesis. One is null hypothesis, another one is alternative hypothesis that may be considered as the research hypothesis also. So what is the difference between the null hypothesis and the research hypothesis? See, the null hypothesis, one of the other hand, is statistically hypothesis that states that any difference is attributable to random errors In other words, the null hypothesis states that there is no true difference, only a random one. For example, I can say here there are two drugs are in the market. One drug is A, another drug is B. A drug and B drug. So now what we are going to see, and in null hypothesis means, see, we are going to say the A drug is not equivalent to B. Okay, so what we can say, see, for example, A may be test, B may be, A may be standard, B may be the test for your drug. So your drug is B means we can say my drug is not biologically equivalent or therapeutically equal to the A. So that means we are telling here null hypothesis means we are not having any statistical significance here. That is the reason we are telling that is called the null hypothesis, which is not at all statistical significance. But in case of research hypothesis, so compared to A and B drug, or it is, we can say like compared to A standard, my drug is good B. So what we are going to do here, the research hypothesis, we are having the statistical significance. See what we are going to keep here, we are having one setup one light level. For example, p value is less than 0.05. If it is more than that, my hypothesis is passed. So what is happening? The research hypothesis is nullifying the null hypothesis here. So there may be error may be there because of failures of the null hypothesis. There are a lot of chances because of the experimental error or statistical error, or really the B drug may be good during the experimental condition. It may be failure. We don't know. So the null hypothesis is very simple. It is not at all statistical significance, but a research hypothesis is significant. It is nullifying the null hypothesis. That means very simple. Why I am telling this? You can take any null hypothesis always. Okay. You have to take any null hypothesis, whatever based on the literature survey or the other things, you take it, you find why there is no significance. What may be the reason? So if you are finding the solution for that, you can develop on research hypothesis. So that is the difference, main difference between the null hypothesis and the research hypothesis. You can see here, null hypothesis, there is no statistical significance. A difference between the sample means of two groups. Any observed difference is the resulting of sampling error, maybe. So that also I am telling, we cannot believe the null hypothesis, maybe due to sampling error. So you can take as the challenge here, because of sampling error and other errors only, this drug is not showing significance because of his null hypothesis. You can, the same thing, you can develop the research hypothesis from this. Okay, if you are proving that, if you are so much of confident on that, you can keep your statistical significance p less than 0.01 also. Okay, understand? So research hypothesis can be developed from the null hypothesis also. That is based on your interpretation and the literature reviews. So okay, research hypothesis, how to write. See, for example, a research hypothesis should name two variables always. Okay, so maybe one to another relationship variables and indicates the type of relationship expected between them. For example, here I given, there is a direct relationship between the concentration of the pollutants and the amount of toxin found in fish. Yes, correct. For example, in the sea, if the pollutants are high, surely the fish will have the more toxins in the another form. There is a relationship. So it must be, hypothesis must be correlated with the two variables. Among college students, there is an inverse relationship between level of free floating anxiety and the ability to form friendship. Yes, of course, because if the students are not having anxiety among the friends group or among the students, the friendship development may be high. So like that, what they're saying, it should be, okay, it should be related at least to two variables. You can relate 2 or 3 variables, but whenever you are making more variables, it is very difficult to develop the objectives. So it should be very simple. You can develop in the maximum 2 or 3 variables interlinking. So a hypothesis should be simple and contain small sentence. That is very important. Say for example, if a comparison is to be made, okay, you are writing comparison hypothesis, the elements to be compared should be stated in the hypothesis. Comparison are induced through the use of words more, less, higher or lower, whatever may be. So you can see here one sentence, low achieving students are more dependent on adults for psychological support. See, what is this? It is an incomplete sentence. See, we have to compare with others. See, low achieving students are more dependent on adults for psychological support than higher achievers. So what it is doing, the average, below average students are dependent on others. So we have to give this motivation, psychological support from teachers or whatever maybe. But a higher achiever students, no need. But this sentence are very, very, very incomplete. So whenever we are writing the hypothesis, it should be fulfilled and it should be completed. And see, hypothesis should indicate what will be the actual studies. That is very important. Not the possible imagination, okay, and the implications of the study or value judgments, whatever you are thinking, that should not be there. It should be actual studies. For example, religion is good for society. Okay, so you are telling, yes, I know, everybody knows. So from this, how to develop the hypothesis? Religious service is inversely associated with the people wrong behavior, correct? If the, see, for example, if the religious service is good to all, the people will have the good behavior. Understand? See, like that, so we have to make our own hypothesis. Okay, it should be related. Also, it should not have the value judgments or imaginations or whatever maybe. It should be actually studies. So that is also, we should be careful. And the name the variables in the order, that is very important, in which they occur will be measured during the reach. See, for example, here, there is a positive relationship between first semester grades earned in college and SAT scores. So what they are telling here, see, when the students are coming in the first semester, okay, those who are having the good marks, it can be correlated with the entrance examination. So that what they are going to say here, the students, those who are getting good marks in the entrance examination, surely will get the good marks in the semester. So here we must understand here, see, which will happen first? First, the entrance exam will happen. Next, only semester exam will happen. So what they correlated here, there is a positive relationship between first semester grades earned in college and SAT scores. It should not be like this. See, always we should write in the order. What happened? Which is happened first? Actually, the entrance examination happened first. Then only the semester happened. So like that, there is a positive relation between SAT scores, entrance examination score, and grades earned in college. So that is the reason hypothesis should be, okay, in order. Avoid using two different terms to refer same variable. For example, don't write to prove, too significant, too so significant in the hypothesis. That means you are deciding there itself. Okay, see, for example, if you are writing to prove so-and-so activity in so-and-so for a disease. So that is not easy. That is to prove too significant, that and all, it should be coming in the objective. It should not come here. Okay, that is also very, very important. So we should not decide everything and put it as the research hypothesis. And also avoid using exact statistical prediction in hypothesis. For example, see here, 35% less bacteria contamination will be found in the air of operation rooms in which the staff wear gloves. See here, what is there? If you are giving like this hypothesis, what is happening? You are giving the answer. 35% less bacteria contamination will found in the air of operating rooms in which the staff wear gloves. See, it should not be like this. See, we should like this. Less bacterial contamination will be found in the air of operating rooms in which staff wear gloves. So we should not give the answer in the hypothesis. Okay, for example, why you can ask, sir, we can ask why we are giving this statement. See, from this statement, again, we will have next, less bacterial contamination will be found in the air of operating rooms in which staff wear gloves. My hypothesis, for example, you are going to develop this. See, which type of bacteria is causing contamination, question mark. That is maybe your hypothesis. Understand? See here, what is that? We should not give any answers or anything. You have to give the general statement. Based on that, your hypothesis should come. Your test is what bacteria is found there. Which type of the species are there? So for example, E. coli or a typhoid, whatever may be. So that is the thing, your test you are going to do for that. See, in a report, hypothesis should flow logically from the narrative that immediately proceeds. A research report begins with an introduction and a literature review, and this should logically lead the reader to the hypothesis. See, here we must understand, before going for any hypothesis or research concept or anything else, we must have the thorough literature review about your subject. Then only we must go, because sometimes whatever you are stating may be wrong also. Okay, because some literatures may oppose that. So we should have the complete report. So we can ask, one literature is supporting, another literature is not supporting. We can go, but the thing is, which is more. For example, 5 reports are saying one report is saying something opposite to that, means we can go for the 5 reports. Here also statistics. Okay, so your concept is supporting by 5 researchers, 5 literature reviews or 10. You can go ahead. If you are seeing any opposing for that, better we can go for the probability. Even we can take it as the opposite. Those who are stated, what may be the reason also we can prove. That is also one kind of research hypothesis we can develop. So what is a research question? That is very important here. See, so far we have seen how to write the research hypothesis. Now, from the hypothesis, how to develop the research question. From the research question, how to go to objective. So, from now we are moving from the research hypothesis to the research question now. See, here, for example, just before I told no, here also less bacterial contamination will be found in the air of operating room in which staff wear gloves. So here you can found which type of the you can from this hypothesis or whatever maybe you can develop your questions. First question, see what type of the bacteria is present. Question. Second question, why this bacteria only is there. Third one, how to eliminate those bacteria also. Okay, so like that you can develop the research questions. From that you can develop the objective. So from your hypothesis you must develop your research question. Interest in a particular topic usually begins the research process, but it is the familiarity with the subject that helps define and appropriate research question for a study. See, very important. It is important to know where the boundary between current knowledge and the ignorance lies. That is very important. Your research question should answer the research gap currently. And the challenge in developing an appropriate research question is in determining which uncertainty is good or should be studied. That is very important. See, for example, you are stating on hypothesis, okay, because from your research gap. So you see, for example, if we are assigning on research question, that should be challenging in developing the answer for the research gap. Okay, so instead of that, simply if you are making research question, that may not be useful for others, and also we cannot create the proper objectives. See, for example, increasing our knowledge about the subject of interest can be accumulated in many ways, and also appropriate method includes systemically. See, for example, just before I told what type of bacteria is there question. So how will you find? So for that you have to develop from the research question, you have to develop one method. See, you have to develop one method. How I am going, how I am going to find those bacteria methodology. Whether we are going for any biotechnological, or if you are going for any microbiological studies, that you have to decide from your research questions. Okay, and also in-depth interviews or focus on groups with patients. For example, if you are doing any clinical, you can develop your research question. Preliminary, you can have one interview, or you can have an interaction with any experts in the field. Okay, sir, for example, we could not develop the research questions, for example. So we can, from research questions, okay, you are having a hypothesis, but you cannot able to develop the research question, means you can go for a random interview with the patients, or you can go with the physician's interview, or you can go with the any expert. From that, you can get the ideas to form the research questions. In-depth knowledge about the subject may generate a number of questions also. That is also, if you are reading more and more, you can develop the more questions. It then becomes necessary to ask whether these questions can be answered through one study or more studies required. You have to decide. And additional research questions can be developed. But my thing is, what I'm telling, instead of developing so much of research question, because it will increase the objectives again, objective will increase the methodology, you cannot be able to correlate finally. So what we must do, the research question very simple and very less. 1 or 3, if you are answering itself, more than sufficient from your study. So basics should be taken into consideration. All primary and secondary should be developed at the beginning and the planning strategy itself. So during the research, we should not develop any research questions. See, from the hypothesis, we have to develop the research questions, then objective, you have to develop the methodology. So during methodology, you cannot come back to the research question again, because if you're coming back to the research question, your objective and methodology again, it will change. Okay, so that should be very, very careful. So before study itself, we develop all the research questions, what we are going to answer, but that should be answer the research gap. And additional question should never compromise the primary question. That is very important. See, for example, you are having the so much of 5 questions or 6 questions. So first question is your primary question, but the first question, and during a methodology or experiments, you are getting another question to answer. That should not compromise or controversy to the primary questions. That is very, very important. Okay, so because it may spoil your hypothesis and study objectives also, it must be kept in mind within scope of one study. The presence of number of research questions will affect the potentially increase the complexity of both the study design and the subsequent statistical analysis also. So too much of research question also kill your research. That is also we must understand here. And also a sensible is to establish a single primary research question around which to focus the study plan. So very simple questions or 1 or 3 primary questions can be able to fulfill your study plan means it is more than sufficient. In a study, the primary research question should be clearly stated at the end of the introduction in the each proposal whenever you are writing. That is very important. See, nowadays, everybody, see, if you are writing hypothesis, then you are going objective. So in between, everybody is seeking the what may be the research question. Means what is the research question going to answer in the objective and also methodology. So always hypothesis creation is very, very interesting thing. But after that, we should not go to objective directly. We can stick on the research questions. Each proposal hereafter, please include the research questions. Based on that, you develop your objectives. And is usually specify the population to be studied. The intervention can be done, and also some of the circumstantial factors also can be estimated using your research questions. See, for example, next we are moving to the research objectives. Okay, so now I don't know. So based on your hypothesis, based on your hypothesis, now we are going to develop your objective. Okay, so how you are going to develop? That is the easiest way. In between, you must create the research questions that may help to develop your objective. So very simple. Objective is the goal intended to be attained. And the very example we can say the sole of objective of her mother trip was to see her children. See, for example, it should be specific. That is very important. For example, see, the object is the goal intended to attain, but for attaining that goal, we should not have particular intention. I have to finish this study. That is very important. See, the objective should have the intention. The person who is framing this objective should not have intention to this complete the study, because if you are having the intention, see, most of the times is happening nowadays. So in nowadays, see, for example, we are forceful to complete the project. So that is the reason the object is we are intention to complete the objective nowadays. So I hope you can understand what I'm telling, and also everybody will agree this. So the framing the objective should be intended to attain the goals, not for our personal or like we have to complete this study, or I have to go for the publications or something. Okay, so I have to complete earlier. So that should not kind of intention should not be there. The intention should be specific to your goal. So also the objective is the state of affairs that a plan is intended to achieve the research hypothesis. Okay, that's what I told now. So the objective will help you to answer the research questions, what you have developed from the research hypothesis. So you can see synonyms, we can keep, some people will keep objective or aim, object or target, goal, or what is your end, like that we can make it here. And research objects writing is a very, very fantastic skill. Also, it is a art. So everybody cannot create the objective, and the objective should come from the discussion from others only. Okay, if you are specifically typing the objectives or framing the objectives, that should be validated by some experts or your superiors or your friends, or it come to the discussion. Because if you are making objective without discussing, if you are developing the methodology, it is keep on going and end with some other thing. If your objective is wrong, so entire your outcome also will be wrong. It may be rejected in end of the day. So research objective formation is very specific to, okay, very specific. Also, it should be, okay, discussed with others, and also it may be consulted with any expert also before going for any research objectives. So better after creating hypothesis, you create your research question. So both the things you go on to discuss with others. So they will tell, okay, what are the research questions problem is there. If it is not fulfilling the gap, it is not answering this. So you may create more research questions. From that, you concise the research question to 3 to 5. Based on that, you come for the objective. Again, the objective can be validated by someone, and you can succeed with this your project proposals. So research objectives are derived from the purpose. They set out what is to be achieved in a research study in specific terms. That is very important. Okay, see, for example, you are doing some other work, but the research objective is irrelevant. We cannot correlate. It should be fulfill your hypothesis. And also, there are crucial in any research since they determine the kind of questions and the procedures to be used in data collection. So, and analysis. So, what they're telling here, so while, okay, see, for example, what is the statistics you are going to do? What is the data collection you are going to do? So what is a methodology you are going to do? That methodology should reflect in your hypothesis, objective. That is very important. I will give you some of the examples also in maybe next slides. Okay, in stating objectives, just like in purpose of the study, you should be non-biased verb should be used. So for example, we should not have the negative objectives here. Okay, you should not affect the others. For example, some people will have, we have seen first objective is controversial to the third objective. Okay, and also it should not have the negative impact on that. It should be non-biased verb should be used. While using the technical terms also, it should be non-biased. To determine, to find out, to investigate, to examine, to explore, to test, establish, to prove, to find significant, and differentiate, inquire, to compare. These kind of words can be always used in the objective. It should be avoided in the hypothesis. It should be avoided in the hypothesis. This is the good verbs to start the any type of the objective based on your studies. And the focus the study, that is very important. Narrowing it down to essential. Your focus to your study. It is not, it should not give the broad objective always. I will give some of the example for the broad objectives also. See, it should be specific. It should be narrow. Okay, to avoid the collection of data which are not strictly necessary for understanding and solving the problems which we have identified. That means, for example, see, if you are going to do from the methodology objective, you are going to do the methodology. So you should not have the methodology. The methodology should not have the irrelevant methods to collection of data, which is not at all related to your objective, research hypothesis or research question. That is the reason in objective forming itself will tell what methodology you are going to do. See, to organize the study in clearly defined parts of phrases, properly formulated specific objective will facilitate the development of research methodology. That is always truth. Will help to orient the collection specific to analysis, interpretation, and utilization of the data. Research objectives are specialization of the ultimate reason for carrying out research. They help in developing the specific list of information needs also. So objective is deciding what you are going to do, what you are going to collect, and what may be your outcome. So outcome may be negative also. We should not, okay, worry on that. Okay, we try to make it positive only. That is also major thing. The very setback in the research nowadays. And in formulating objectives, care must be taken, especially to state the objectives that are very clear, well written, and precise. Make objectives specific, significant, realistic, and achievable. That is very important. Somewhere it is not achievable means nobody will listen to that project. Okay, and based on your feasibility and what is your facilities available. Ensure that objectives follow logically from the statement of need and address the problem. That is very important. So objectives should address the problem which you have raised in the research questions. Make objective fall within the range of result which are expected to be achieved within the limit time of the time. Okay, what is the proposed time? So don't put so much of the things, it may not favor for you, especially it may lead to a lot of money wastage and human resource. Okay, a lot of problems will come. And also state the objectives as far as possible in terms which allow measurement or at least observation for their achievement. And objective should be chronological. That is very, very important. See, first sentence, what they're telling, the objective should be achievable in terms of measuring the data or collecting the data or better observation. But in the chronological order, that means, for example, I am giving one example. First, you are starting from one plant, means first objective should be to collection of the plant. Next should be to prepare the so-and-so extract. Third one is to fractionation. Fourth one is to isolate the some of the common from the fractionation. Next, to characterized by using IR, NMR, mass. So then in vitro study. Then like that, it should be chronological order. Some people will write the first itself in vitro study and they will go for the extraction. So that is nowhere it is relatable. Remember once more, then the objective of the research project summarize what is to be achieved by the study, and it should be closely related to the statement of the problem. That is very important. So whatever the problem you have created, okay, in the research hypothesis and research questions, okay, so when you are making the number of objectives, okay, all the objectives should fulfill the statement of the problem. Everything come back to the one place. That means what is the origin of your research question and the problems. So yeah, all the things should answer the your research question and the hypothesis. That is very important. All the objectives. So so in case, okay, so if you want to develop so much of objectives. So for example, format used in the stating objectives, better you give first general objective. So for example, to isolate the so-and-so common from this extract for the Alzheimer's disease. That is maybe your general objective. Okay, to isolate, for example, to isolate flavonoids from methanolic extract of, okay, so Empirica officinalis for Alzheimer's disease. This may be your general objective. You know what may be the specific objective for that. So from that, first I told to collection, to extraction, to fractionation, to isolation, to characterization, then chemical characterization or whatever maybe, then your in vitro study, then acute toxicity, then in vivo. Like that, the objectives may be more, but everything you should fulfill your general objective. General objective can come from the research questions and also hypothesis. So that is very, very important. So smaller logically connected parts of the general objective, it should be connected. Your general objective connected to the specific objective. They are the specific aspects of topic we want to study within the framework of a study. Specific objective should systematically address all the various aspects of the problem and the key factors that are assumed to influence or cause the problem. They should be specify what we'll do in our study, where, for what purpose we are developing. That is very important. So the general objective may be broad, specific objective to address the general objective. The general objective, it's formed from, okay, your research hypothesis and questions. And for example, here, again, general objective to identify the antiviral chemical constitutes in the, okay, so some of the plants, flowers. So specific objective, first to carry out the solvent extraction of dried flowers and to isolate the constitution of the extrude extract, to determine the antiviral activity of the crude extract, and to determine the structural formula for this active compound. See how much they have done. Understand? See, one specific objective, it came to the various, general objective become the specific objectives. See, and now some of the examples I gave from my study. See, to design synthesis and evaluation of novel glidosomes for Alzheimer's disease. Activation of PGC1 alpha signaling via PPAR gamma receptor is my expected mechanism. For example, general objective. So I am not giving the lot of objective here. I given some of the specific objective here. See, because to study the PPAR gamma binding affinity of synthesis compounds, In vitro neuroprotective evaluation of synthesis novel gliders in suitable in vitro model to evaluate the neuroprotective effect on cognitive enhancement activity of synthesis gliders in suitable in vivo models. See, while I made this presentation in the panel, especially in various funding agencies, the members were objected me. Okay, and also they shown the specific, the objective is not good. Why? And it is not specific. It is come for the open discussion now. So anybody can find out what may be the problems, why the members have objected this specific objectives, what may be the reason, how it can be modified. So please come out and we can have a discussion, please. So anyone or anybody can have a discussion, then I will give the answer what I have changed in next presentations, what is the mistake I done in the first presentation, why they have opposed that. Anybody, any participants? Anybody is there? Any idea? Okay, so it's fine. I will tell the answer. See what happened. The refined objective. First, we can see to study the PPAR gamma binding affinity of synthesis compound. Just I gave. Next, to study the PPAR gamma binding affinity of synthesis compounds by in vitro protein binding assay. What methodology I am going to follow and what type of kit we are using Lantha screen comparative binding assay kit. This was missed here. Second thing is in vitro neuroprotective evaluation of synthesis novel gliders on amyloid beta peptide intoxicated cortical neuronal primary culture, but I gave insuitable in vitro model. That is also objection. Here also you can see in vivo in the suitable animal models. That is also wrong. So next, the synthesis glidosomes in intracerebroventricular administration of beta amyloid in rat model. So like that, the objective should be specific. You should tell what you are going to do, where you are going to do, how you are going to do, by using what, which model, everything they must understand from your objective, because most of them will not read anything in the methodology. Sometimes if your objective is good, they will go for the project recommendations. So finally, an objective is a clear statement of something that needs to be accumulated over a period of time. Objectives are specific, states exactly what you need to achieve. Measurable, includes the quality or quantitative measures. Achievable or agreeable between you and your supervisor or researcher reviewers. And realistic and relevant, can be challenging but must be achievable. And time bound with a clear date or time scale should be there. So we cannot do the long time. So with this, I am concluding my talk. So thank you once again for your opportunity given, and especially the organizers of the PhD College of Pharmacy. Thank you. So any questions are welcome. Any discussions?
Thank you, sir.
Yeah.
Dear delegates, if you have any questions, you can post it in your chat box or unmute and ask directly to sir so that it can be clarified. So I think there is no questions, sir. And
I think I feel that everybody available
are available, sir. Really, your speech was enlightened, and the way how and why, how to formulate objectives, everything was very clear, sir, for upcoming research scholars. And thank you very much for your valuable time spent for us, sir. And participants will have a short break of around 15 to 20 minutes. We'll start at 11:30. Thank you, participants, for your kind cooperation.
Thank you so much, sir.
Yeah. So shall I wind up here?
Yeah, yes, sir.
Yes.
We can wind up. We can wind up.
Okay, but thank you. Huh?
Okay,
Thank you, sir.
So participants
you, sir.
will join by 11:15.
Sir, we are joining by 11:15.
Yes, ma'am. We'll join by 11:15.
Okay, sir. Thank you, participants. We'll join by 11:15.